CRISPR is as good a name as any. It used to be recombinant DNA or biotechnology. Now it's CRISPR, but it's a cluster of molecular tools that we discovered in nature through basic science. And then we started combining them various ways. So for example, you can grab a deaminase from transfer RNA and graft it onto a phage attack machine and disable the nuclease. And now you've got what's called a base editor, which is not found in nature.
Hello everyone, I warmly welcome you back for another session in the series of interviews organized by Department of Chemistry, University of Colombo, where we talk to eminent scientists all around the world to get an insight into their scientific journey by walking through their experiences. I am Udit Tilanka, a fourth year undergraduate at the University of Colombo. With me today, I have Shanika Sandeepani and Hidji Selva as my fellow co-host. So over to you, Shanika.
Thank you, Vidu. So let's welcome our eminent guest for today's interview, Professor George Church, who is a professor of genetics at Harvard Medical School and a professor of health sciences and technology at MIT. Professor George is widely recognized for his innovative contributions to genomic sciences and his many pioneering contributions to chemistry and biomedicine. In 1984, he developed the first direct genomic sequencing method, which resulted in the first genome sequence. He helped to initiate the Human Genome Project and the Personal Genome Project in 2005. Professor Church, it's truly an honor to have you here with us today. So how are you today?
I'm fine. I'm glad to be here.
We are glad to hear that. So to start off this session, I'll invite Hiruni to ask some questions from our eminent guest.
Thank you, Shanika. So, Professor, before we move on to talk about your research and career, please tell us about your early life, education, and what drove you to follow biology and what made you passionate to pursue your work in the field of genetic engineering.
Well, my early life, I did not have really any access to science. They didn't teach it in school, and I didn't know anybody who was a scientist or engineer. That got corrected when I moved from Florida to Massachusetts when I was 13 years old. And I immediately became fascinated mainly with biology and chemistry, but a little bit in physics and computer science. And I started programming immediately, finished all the chemistry courses they had to offer, the most advanced ones. And then I got access to the lab as independent study, meaning that was the only one in the lab at the time, like on weekends and evenings. And that was quite an experience to be trusted with being able to be alone in a chemistry lab. And I didn't do any damage as far as I know. And then, but at that time when I started my undergraduate the next year, I immediately sought out some researchers to do both computational work and then soon after chemical crystallography and computational chemistry, mostly focusing on transfer RNA. And then since transfer RNA was a genetic molecule, arguably the most important genetic molecule, it led me into all the rest of the things that we're talk about. But it started with crystallography, really.
Few years ago, you and several other scientists initiated the Genome Project, right? That focuses on the development of technologies for the synthesis and testing of genomes of many different species, including the human genome. So can you please elaborate on this and tell us about its future prospects?
Well, in 1984, it was most of the discussion was focused on the feasibility of doing one human genome, meaning not even in your cells, you have two human genomes, one for your mother and one for your father, but we were going to do one, which would not be clinically useful, but nevertheless, that was our goal. And somebody came up with a number of $1 a base, which would be $3 billion, which I thought was completely unacceptable. Nevertheless, that went forward, and we did eventually spend more than $3 billion and did not finish the genome, even the one genome. And then, fortunately, after that finished in 2004, my group and others started a more rapid project to develop new technology. The first project didn't really develop new technology between 1984 and 2004. But the second project from 2004 to say 2008, much faster, much cheaper, and it brought down the price by about 20 million fold. So now instead of $3 billion for a poor genome, it's $300 for high quality clinical genome due to next generation sequencing. And you asked beyond that in the future, I think we're moving toward three-dimensional imaging with sequencing, where every image dot in a microscope has a chemical tag that tells which protein, RNA, or DNA is at that 3D coordinates. That's one kind of sequencing. And the other kind of sequencing will be highly portable, wearable sequencing will be very inexpensive, and everybody will have it like on their cell phone. And that will help with prevent pandemics and smaller medical issues.
That's quite interesting. Let's hope the Atlas of the Human Genome will revolutionize medical practice and biological research into the 21st century.
Okay, Professor, following on that, you are world famous for your project on reviving the woolly mammoth after thousands of years since they disappeared from the Arctic tundra. What is the purpose behind that, and how is it going, Professor?
So the main purpose is to develop new technologies that can be useful for saving endangered species, bringing them back from the brink of extinction. And that can include getting chemically synthesizing a diversity of DNA alleles, that is, variations on a DNA sequence, building them up from the computer into real DNA in living cells in endangered species, and then reintroducing those into the wild to make them more well adapted to the modern environment, to make them more diverse if diversity is threatening their survival. So that's the main objective. The secondary objective is specifically, that's a general topic. A specific application is for the elephant, to make them to restore cold resistance. Both the Asian elephant and the African elephant are endangered species, and the Borneo elephant and forest elephant. There's a few endangered. All elephant species are endangered, and we would like to give them. One of the reasons they're endangered is because they're in conflict with high human population densities. So we'd like to give them access to places where human population density is nearly zero. And so that's and then the third reason and final reason is that once they have access to the Arctic, which is such a place with very low human population density, then they can help restore the Arctic to a time when it was it was better at sequestering carbon than it is today.
Okay, Professor, that's pretty cool. Shanika?
So let's jump right into the interesting topic of age reversal. Can you tell our listeners a bit about using genetic engineering to cure aging, and what is fascinating about age reversal?
So we use the term aging reversal rather than longevity because it takes too long to do a proper longevity clinical trial in humans. Really, there's so much variation in how long people live that you really have to do a 30-year trial to be accurate about that. But aging reversal can happen in weeks. So it's much more like a conventional drug, and quite likely the sort of drugs that produce aging reversal will also produce longevity, but we just don't need to convince the FDA or equivalent agencies around the world. That said, that there are aging involves multiple pathways, some of which interact in various ways, somewhere around 9 or 10, depending on how you count cancer as part of aging, lots of age-related diseases. And so you have to make sure you're getting all of them, because if you just get a few of them, then you might extend life or health by 3 years. But if you get all of them, the hypothesis is then you might get true aging reversal. And so we think that getting that many at once is probably most easily done by gene therapy. Gene therapy is the same kind of process we used in the vaccines recently, and it can be as little as $4 a dose if you have a large population affected, which is the cost of the AstraZeneca COVID vaccine, which is an adenovirus wrapped around double-stranded recombinant DNA. So we think that we can deliver multiple genes as a generic aging reversal that will affect most, if not all, diseases that are affected by aging.
Great. If it works well, then definitely it would be a giant step forward in the field of genetics. So wish you the best of luck, Professor.
I should just add that we've published 3 papers on mice, 3 different gene therapy, 3 different combinations of gene therapy, and now we're doing clinical trials in dogs for a veterinary product, and that's looking good. And so then we'll start human clinical trials soon. So it's important to talk about not just the plan, but the progress so far. So thank you.
Fantastic. So moving on to the next question. I know the question, what is CRISPR, may be the most asked question from you in interviews like this. But I will take a little detour and ask you, why is CRISPR? What makes it interesting?
Well, I think CRISPR is really an icon. It's like it's not interesting for the thing itself. It's interesting for all the things it represents, and it represents our growing ability to read, write, and edit DNA. You can't edit DNA without being able to read it, both before and after. You can't refine the methods for editing without reading. So I think that it's a combined revolution, and it's not restricted to CRISPR. There are quite a number of ways of doing gene therapy, of genome engineering of agricultural species and so forth. But CRISPR is as good a name as any. It used to be recombinant DNA or biotechnology. Now it's CRISPR, but it's a cluster of molecular tools that we discovered in nature through basic science. And then we started combining them various ways. So for example, you can grab a deaminase from a transfer RNA and graft it onto a phage attack machine, and disable the nuclease. And now you've got what's called a base editor, which is not found in nature. So it's a good platform. All of these things provide mutual platforms for developing new technology that goes far beyond nature.
The gravest concern regarding CRISPR is that it enables germline edits that they get passed on to future generations. In other words, you are permanently changing the human species when you do that. What is your idea regarding this, Professor?
Well, I think that germline manipulation of humans is not particularly useful. A good, solid use case has not been described. It is also difficult to prove the same way I was saying about longevity is difficult to prove because the clinical trials take too long and are therefore hard to, they're expensive. It's also true for germline, where you really should do the clinical trials over 1 or 2 human generations. So you're talking about something even longer than a longevity study. So those are the 2 problems with germline. There are some advantages to germline, which is you get complete delivery to every cell in the body if you want it, which you don't get from somatic gene therapy. You also can get very thorough testing of something. So, and so that said, the way that we're getting all the advantages of germline without the disadvantage, the ethical and safety issues is by germline manipulation of pigs. And then the pig organs can be transplanted into humans that need the organs, and those organs can be enhanced. So they're actually resistant to pathogens, they're resistant to aging and to cancer. And so they're actually better than the organ that they're replacing, but you're only doing it in cases of, initially of cases of medical need. So I think the advantage of germline can mostly be accessed by that way. It's not appropriate to use germline for dealing with, say, rare genetic diseases. Rare genetic diseases are have been proven to be easily addressed by genetic counseling. In the communities that use genetic counseling, they've almost completely eliminated Tay-Sachs and 9 other genetic diseases. So we know that works. It's very inexpensive compared to any kind of germ therapy, any kind of therapy in the orphan drug category, meaning for rare diseases. Those typically run millions of dollars per patient per lifetime.
Professor, as we know, our epigenome plays a key role in expressing the code of life inside us. But still, we are in the initial stages of tapping into this aspect. What are your thoughts on using genome editing tools in editing the epigenome?
So the epigenome is not as mysterious as it was a few years ago. We are now using it routinely. We understand it well enough that we can now engineer it. And examples of what we're doing to engineer it is we're building. We now have almost 300 recipes for how we can take a stem cell and turn it into almost any cell in your body. And it looks like we can make any cell type, meaning any epigenomic cell type. So we can make liver cells and kidney podocytes and endothelial cells on your capillaries and neurons and glia and so forth. So that's one example of epigenetics. We can also use epigenetics in plants where it's actually inherited from generation to generation as if it were a mutation, but it's really just changing, say, the methylation status of the DNA or the proteins bound to the DNA.
As we all know, we are still living in the middle of a pandemic. Is there any research going on regarding COVID-19 that you or your colleagues are involved in that you would like to share with us today?
Yes, I mean, as soon as we heard of COVID-19, my lab started doing research on several aspects of it, including drugs to treat it, vaccines to prevent it, and diagnostics to monitor not just the presence or absence of the virus or the presence or absence of antibodies in your body to the virus, but also look for development of resistance to drugs and vaccines. And we try to do all of this in the context, not just of this pandemic, but of many pandemics to come, because this is not the first or the last. We had before SARS-CoV-2, we had MERS, we had Ebola, we had swine flu. We had many of these are zoonotic diseases that come from our lack of respect for the wild environments where we either destroy the forests or we eat wild food without proper respect. Anyway, we need preparedness for this, and it needs to be affordable so that everybody in the world is protected, because even if you're a rich person, you should care about everybody, because even if for selfish reasons, it will come back to haunt you. So I think that's been an emphasis of my lab, is to bring down the cost of DNA sequencing and therapeutics. And so that's why it's time to celebrate that the vaccines are as low as $4 a dose for the AstraZeneca vaccine. And it's also, it's a good time for thinking about ways that we can keep up with evolving strains. This one did evolve. The Delta is the most famous variant, but it involved many variants, and it's probably not done yet. This may be something that is becomes endemic to the planet, that we constantly are exposed to this. In principle, we should be able to eliminate it because, like smallpox and polio, it is fairly human specific. But I think we need these powerful tools made affordable.
Following on that, we would like to know a little about your research plans for the future. What are you planning to do with this repository of genome editing tools available now?
So many of the things we do, we call multiplex editing. So we're not changing just one gene at a time. We have to change many genes, and sometimes in naturally occurring populations. So one of the things that we proposed were a set of ways of set of safety regulations and technologies that allow one to safely inhibit the spread of pathogens. For example, Lyme disease via ticks and malaria via mosquitoes. And so we hope that that can be done in a very cautious manner. But that might involve multiple genes to protect, let's say, the mosquito from the Anopheles mosquito from the parasite malaria. And then another one is engineering pigs. We had to make 42 changes in the pig genome so that they would be close enough to human immune surveillance so that we can get immune tolerance and do organ transplants. Because it's not just that there's a worldwide shortage of organs, but it's also that an opportunity for making the organs stronger than you would get from a human. And then the third and final example of using multiplex editing is we are making organisms resistant to all viruses by changing the genetic code. This requires somewhere between, the smallest number so far is 300, and probably the largest we have planned is 400,000 edits in order to change the genetic code to make industrial microorganisms, plants, animals, and human cells resistant to all viruses, even viruses we haven't seen before. So that recoding is probably one of our biggest chemical projects because we're changing 6 billion base pair genome in up to 400,000 places. Our record so far for multiplex editing is 24,000 edits to human stem cells, all done by deamination of A's to G's. So the nitrogen, the exocyclic nitrogen, and the adenine is changed to an oxygen, making it an inosine, which has been copied into a guanine. So that chemical reaction is done 24,000 times in one cell, and then that cell can be clonally propagated as a stem cell. So that's multiplex editing. I think that's one of the most important categories for editing right now.
Wish you success in all your projects, Professor, and hope everything goes well.
Thank you.
Okay, the next question. We are gradually marching towards a novel space age. Humans will probably evolve into an interplanetary species in the coming 100 or so years. What will the future of genome editing look like in such a futuristic landscape? We would love to hear your thoughts and predictions on that.
Right. So in the human population has evolved to live in open fields and woods for the most part, not in cities and definitely not in space. And hence we might need therapies or genetic manipulations that help us live in those confined spaces, both for psychosocial reasons, but also low gravity, infectious diseases that are particularly problematic in enclosed spaces. The reason we have these epidemics in the wintertime, like the flu, is because everybody's in these closed spaces. That'll be even more so as cities get denser and as spacecraft get denser, or colonies for that matter. And radiation. So the Earth has a much better protection from radiation than does a spacecraft or, say, a Mars colony. And so radiation, low gravity, psychosocial, and pathogens all require a fresh look. And I think we can do this with somatic gene therapy on every newborn. It'll be as if it were germline, but it will be done at a very young age without germline manipulation.
That's very interesting. So the final question, Professor, what advice would you like to extend towards undergraduates and budding scientists, especially here in Sri Lanka, who are striving to do impactful researches in those fields? We would love to hear that.
Right. So it's actually a wonderful time to be a scientist anywhere in the world, really. It is a time of revolution. And the nice thing about this kind of peaceful revolution is it levels the playing field. Every few years, everybody has to learn all at once. And so you're never really behind, or you're no further behind than anybody else is. So that's one good thing. The cost, part of that revolution is the cost coming down. And so we're seeing citizen science around the world. I've been involved in some of the first 3 citizen science movement, biohacking part of citizen science. And I think that's something in the form of iGEM. We have hundreds of sites around the world where students can do cutting edge biotechnology development. Some of those later turn into commercial successes. So we can try to continue to bring down the cost and make it, and also the instruction is available much more readily on the internet now. The travel becomes less significant. Like right now, we're traveling at the speed of light. And we can communicate across vast distances. So I think all of that results in a much more hopeful scenario for students around the world rather than just in elite centers. It still is worth visiting an elite center for some short period of your career if you can, but it's not nearly as essential as it used to be.
That brings us to the end of today's session. Thank you very much, Professor Church, for taking time out of your busy schedule to join with us and for sharing your valuable knowledge and experiences. It was quite inspiring. We enjoyed every minute of this interview. We wish you all the best in your future endeavors. That's it for this session. Thank you for joining us and stay tuned for more.
Thank you very much. You've been excellent interviewers, and it's a very intelligent conversation. Thank you.
Okay. Thank you, Professor.